For millennia, extinction has been the end of the story, a silent closing of a chapter in the book of life. But what if science could offer a second chance to lost species? Welcome to the world of de-extinction. Across the globe, scientists are piecing together the genetic blueprints of vanished creatures, hoping to breathe life back into them. These efforts are more than scientific curiosities. They challenge our role as stewards of the planet. Success could reshape ecosystems and help combat the extinction crisis. But the path is fraught with technical and ethical challenges. Join me as we explore the science, the people, and the quest to reverse extinction, a journey that could change our understanding of life itself. Our journey begins in the vast, windswept expanse of the Siberian tundra, a place where the land stretches endlessly beneath a pale sky, and the air carries whispers of an ancient past. Here, beneath layers of ice and earth, the remains of the woolly mammoth lie astonishingly well preserved in the permafrost, offering a direct link to a world that vanished thousands of years ago. These colossal Ice Age giants once roamed these plains in great herds, shaping their ecosystem by knocking down trees, fertilizing the soil, and maintaining the grasslands. Their disappearance, brought on by a changing climate and human hunters, transformed the land forever, leaving behind a colder, less diverse world. Today, scientists are inspired by the idea that reintroducing a mammoth-like creature could help restore the ancient mammoth steppe, a vast, grassy ecosystem, and even play a role in slowing climate change by preserving permafrost and reducing greenhouse gas emissions. True cloning isn't possible, no intact mammoth cells have survived the millennia, so researchers are turning to the revolutionary tool of CRISPR gene editing, which allows them to precisely modify DNA. By carefully splicing key mammoth genes, those responsible for thick fur, fat storage, and cold resistance, into the DNA of Asian elephants, they hope to create a cold-adapted hybrid, a so-called mammoth phant. The process is incredibly complex, involving the creation of lab-grown embryos, the use of advanced gene editing techniques, and possibly even the development of artificial wombs to nurture these embryos to term. The goal isn't to bring back a perfect replica of the woolly mammoth, but to engineer a functional proxy, an animal that can survive and thrive in the harsh Arctic environment and help restore balance to the ecosystem. Early experiments have shown that mammoth genes can function inside elephant cells, a small but crucial step forward, hinting at the possibilities that lie ahead. The challenges, however, are immense. Scientists must solve complex genetic puzzles, address animal welfare concerns, and navigate the deep ethical questions that come with reviving extinct traits. Yet the vision remains powerful and inspiring. Herds of mammoth-like creatures once again roaming the northern landscape, reshaping the tundra as they did millennia ago. It's a testament to human curiosity, ingenuity, and our enduring desire to reconnect with and restore the wonders of our planet's lost past. Some extinctions happened before our eyes, moments in history when entire species slipped away, leaving only memories and museum specimens behind. None more poignant than the story of the passenger pigeon, a bird whose fate became a symbol of both abundance and loss. Once numbering in the billions, their flocks would stretch for miles, darkening North American skies for days at a time, so vast that the sun itself seemed to disappear behind their wings. Hunted relentlessly for food and sport, and deprived of their forest homes by rapid deforestation, they vanished in a matter of decades, a collapse so swift, it stunned the world. The last of her kind, Martha, died in 1914 in a lonely zoo cage, leaving a gaping hole in the eastern forests, and a haunting lesson for generations to come. Passenger pigeons were more than just birds. They were keystone species, shaping entire ecosystems with their sheer numbers, dispersing seeds, and nourishing countless other creatures. Their absence is still felt today, a silence where there was once thunder, a missing rhythm in the forests that once echoed with their calls. Now, a century later, the Great Comeback Project dares to imagine a future where these birds return, using cutting-edge gene editing to modify the DNA of their closest living relative, the band-tailed pigeon, with passenger pigeon traits. The process is painstaking, creating chimeric birds that can eventually produce pure passenger pigeon offspring, each step a blend of hope, science, and patience. It's a slow, multi-generational effort, rebuilding a species, gene by gene, with no guarantee of success, but with the promise of possibility. The ultimate goal is breathtaking: to return flocks to the wild, to see the skies alive again, and to restore the lost ecological processes that once shaped entire forests. Even a small flock taking flight would be a powerful symbol of hope, a sign that healing is possible, even after unimaginable loss. This is about more than a bird. It's about healing a wound we inflicted, about learning from our mistakes, and about the responsibility we share to mend what we have broken. De-extinction here is not just science fiction. It's a quest to restore balance to nature, to bring back the echo of a billion wings, and to remind us that our actions, for better or worse, shape the world we all share. In Tasmania, the thylacine, known as the Tasmanian tiger, haunts memory and myth. Its striped back and dog-like face are etched into the island's collective imagination, a symbol of both wildness and loss. For generations, stories of the thylacine have been whispered in the forests and towns, blurring the line between fact and legend. This unique marsupial predator was hunted to extinction, the last dying in 1936. Driven by fear, misunderstanding, and a government bounty, people once saw the thylacine as a threat to livestock, rather than a vital part of the ecosystem. Its disappearance was not just the loss of a species, but the erasure of a living piece of Tasmania's identity. Its loss left Tasmania's ecosystem unbalanced and its people with deep regret. The forest grew quieter, and the absence of this apex predator changed the delicate web of life. Many Tasmanians still mourn what was lost, and wonder what might have been if the thylacine had survived. Now, scientists at the University of Melbourne are working to bring it back. Their ambitious project is fueled by a mix of hope, cutting-edge technology, and a desire to right an old wrong. Using DNA from a preserved thylacine pup, they're editing the genome of its closest living relative, the dunnart. This tiny marsupial, though unassuming, holds the key to reviving a lost species. CRISPR technology will transform dunnart DNA, piece by piece, into that of the thylacine. Each genetic change brings the project one step closer to recreating an animal unseen for nearly a century. The embryo, once created, will be grown in a surrogate dunnart's pouch. This process mimics nature, using the dunnart's unique biology to nurture the thylacine's return from extinction. Marsupial reproduction offers unique advantages: short gestation, tiny embryos. This makes the complex process of de-extinction more feasible, allowing scientists to monitor and support the developing animal at every stage. The project is daunting, requiring millions of genetic edits. Every step is a challenge, demanding patience, precision, and a willingness to venture into the unknown. But the prospect of seeing a thylacine in the wild again is electrifying. It sparks the imagination and inspires hope, not just for Tasmania, but for conservation efforts worldwide. It's a chance to atone for past mistakes and restore a lost predator. The thylacine's return could help heal old wounds and remind us of our responsibility to protect the natural world. The thylacine's return would be a symbol of hope and scientific possibility. It would show that with determination and innovation, we can undo some of the damage we've caused. Sometimes legends can walk again, and when they do, they remind us that the future is still unwritten. Not all de-extinction stories are about the future. One has already happened with bittersweet results. The Pyrenean ibex, or bucardo, was hunted to extinction. The last Celia died in 2000. Scientists preserved her DNA and in 2003 used cloning to bring her back. Out of hundreds of attempts, only one clone was born, a historic first. But the newborn survived just 7 minutes, succumbing to a fatal lung defect. This fleeting return proved that cloning extinct animals is possible, but fraught with challenges. High failure rates and genetic defects reveal how much we still don't understand. The story of Celia's clone is both triumph and tragedy. It's a reminder that resurrection is only the first step. De-extinction is not just about bringing back life, but ensuring it can thrive. The bucardo's brief revival is a lesson in both the promise and limits of science. Sometimes, the past can only visit us for a moment. No animal is more synonymous with extinction than the dodo. Native to Mauritius, the dodo was perfectly adapted to a world without predators, until humans arrived. Within a century, hunting an invasive species wiped it out. For years, reviving the dodo seemed impossible. No soft tissue survived the tropical climate. But advances in ancient DNA allowed scientists to sequence its genome from a preserved skull, its closest living relative, the Nicobar pigeon. The plan used gene editing to modify Nicobar pigeon DNA, recreating dodo traits. The process is long and complex, requiring chimeric birds to produce dodo-like offspring. The dodo project is ambitious, starting from a fragmented past. But bringing the dodo back would transform a symbol of loss into one of restoration. It's a chance to rewrite the story of extinction. Not all de-extinction requires high-tech labs. Sometimes, it's about selective breeding. The quagga, a zebra subspecies with faded stripes, vanished in the 1800s. Genetic analysis revealed it was not a separate species, but a variant of the plains zebra. The Quagga Project in South Africa set out to breed back the quagga by selecting zebras with quagga-like traits. Over generations, these zebras, now called Rao quaggas, closely resemble the original. They're not genetically identical, but visually and functionally similar. The goal: re-establish herds in their ancestral habitat. This project shows that sometimes, lost traits can be revived through careful breeding. De-extinction can be as much about patience and observation as technology. Sometimes the past is hidden in plain sight. In Queensland's rainforests, the gastric brooding frog had a unique way of raising its young, inside its stomach. Both species vanished in the 1980s, victims of habitat loss and disease. Scientists realized the frog's biology could have revolutionized medicine, offering clues for treating stomach disorders. The Lazarus Project aims to bring it back using cloning. Preserved tissue samples provided the genetic code. Scientists implant the extinct frog's DNA into eggs of a related living species. Early experiments have produced dividing embryos, though not yet full tadpoles. These results prove the extinct DNA can be reactivated. The project is a quest to restore a biological marvel and unlock secrets of evolution. Sometimes, the most extraordinary stories are hidden in the smallest creatures. De-extinction offers extraordinary promise and profound questions. The ability to revive lost species could reshape ecosystems and our role as stewards, but this power comes with responsibility, ethical dilemmas, animal welfare, and unpredictable consequences. De-extinction is not a substitute for protecting what we still have. It's a tool, not a cure-all, a last resort, and a lesson in the value of life. The science is advancing fast, and the debate about which species deserve a second chance is just beginning. So if you could bring back any animal, which would you choose?